I want to first thank the organizer. Thank you, Mariana. Thank you, Michael, for hosting me for U-RIG Hour. I was supposed to present 20-minute talk, so please forgive me if you will have some extra time for afternoon on Friday. But first, before I delve into the microbiology presenting today, I do want to set up saying that we are from Northern Alabama, and we are from University of Alabama in Huntsville, which is known as previously as a Rocket City, right? So just to make sure that you know where we are, where to find us. Let's talk about urinary microbiome. Everybody's familiar with that, but I would like to give more introduction to kind of bring our strong motivation why we are studying microbiome interaction in this niche. So we are interested in the urinary tract infections, and in particular, because they are usually considered benign infections and caused by enteric bacteria, that the paradigm coming from the GI tract, and the main causative uropathogens considered to be uropathogenic ecology, or otherwise called UPAC, Klebsiella, and the Enterococcus species. And urinary tract infections, while they're considered benign, they are plaguing millions of people worldwide. Again, this audience probably well familiar with that, but just let's bring everybody to the same page. They account for over 15% of all antibiotic prescriptions in medical field, if you're not discussing other antibiotic applications. And unfortunately, UTI is accounting for a big chunk of hospital-acquired infections, in particular because once you're in the hospital, you're often not feeling well and you frequently receive catheterization. And there is a big portion of research done on catheter-associated UTIs. And these infections from hospitals, as well as community-acquired UTI, are frequently now becoming drug-resistant or multi-drug resistant infections. And these are really hard to treat, especially because the only dominant way to treat UTI infections is actually antibiotic applications. While some of them might be resolved quickly, but if you get MDR-resistant infection, that is really hard to treat. The only option, some people eloquently put it as a antimicrobial-resistant AMR and UTI is an ignorable problem in healthcare. But last Halloween, to scare students and basically drive the point, I got a few more scary slides, and I'm going to show the numbers just to kind of try to convince you. When I was starting the group back in 2018, many of us refer to the predictive study or estimates were done by UK Antimicrobial Resistance Review back in 2016, predicting that in few decades, by 2050, we'll have over 10 million a year deaths associated with drug-resistant infections, all of them, right? And it was daunting, it was scary, but it was a prediction, and people tend to think that it will never happen, and the morbidity or mortality associated with drug resistance would not exceed cancer mortality, right, or other dominant killers. And then just during the fall, like kind of slowing down of pandemic, the famous now Lancet study was released, which actually took a step and calculated all hundreds of country data from 2019, so pre-pandemic time, and estimated the deaths which can be associated or even directly attributed to antimicrobial resistant bacteria. And here's the graph that was not presented by those for those newspapers, which of course picked up the main information, driving home the point that antimicrobial resistance is indeed unignorable and it's causing millions of deaths annually, right? And it was, again, quite a few years ago. And you can see 2019 estimate puts it at about 5 million. But if we consider, as I'm a lecturer, I usually lecture too much, probably in my general microbiology class. People are so used to high numbers, and it's hard to grasp the high numbers. Therefore, I put together then current slide from the existing dashboard for coronavirus from World Health Organization. It's one of the only few which is still running up to date. And I was putting the cumulative deaths associated with SARS-CoV-2 virus infection to about 7 million, right? And this is accumulated over several years. And it's a global pandemic. So if you compare the numbers of 5 million versus 7 total over several years, we can imagine that pandemic of antimicrobial resistance is here, and people actually making nice movies and trying to bring awareness that antimicrobial resistance is a huge problem. And that's, I'm showing my favorite cartoon here, saying that objects such as AMR resistance are actually much closer than they appear. They're already here and they're increasing. So if we consider from perspective of urinary tract infections, so uropathogens, we can just basically show the two dominant uropathogens I just showed, E. coli and Klebsiella, are within the first three top drug-resistant pathogens, killing hundreds of thousands of people a year. And if you consider other uropathogens, they're not really much further, and they are a big problem. So we also know, and in this community in particular, I should still put an exclamation mark that urine is not sterile. We are well aware for last over a decade that there is a urinary microbiome, so community of microbes occupying different parts of urinary tract of humans, and that it contains a variety of bacteria and other microbes. And so now I'm just deleting the exclamation point. We need to study and understand how it interacts with the host, and of course, how it may or may not affect urinary tract infection development. We collaborate with the great group of clinical and bioinformatics researchers in trying to figure out the initial steps, how this microbial niche develops throughout the life of the host. And the hope is that once we consider this microbiome as ecological niche of microbes, how they interact and how they develop and affect the host, we may be able to develop diagnostic tools, so figuring out which of the host or which of the individual will go to develop condition of recurrent UTI or will be prone to develop this condition, and those who would be remaining healthy in urinary health and other conditions, right? But for that, we need to know basically how the occupation of the niche starts, how it proceeds, and what happens within the niche. But up until now, the tens of studies developing they are studying the composition aspect. So basically cataloging who present in a niche and how it associates with different health conditions. So to trying to take it a little bit further, we're trying to add studies. So in this particular study, initial characterization of the pilot cohort of pediatric patients and trying to understand who is occupying urinary tract of children. That study is done in collaboration with Dr. Kelly from Duke University and Dr. Carson from OSHU. We looked at a small pilot study cohort of kids undergoing VCUG procedures, so for medical reasons. And we try to use different methods. So try to bring a little bit more taxonomical resolution to figure out who is occupying urinary tract. So what we observed that indeed, like now several other studies reported, both boys and girls contain a polymicrobial urinary community. So we have different number between individuals of bacterial species occupying this niche. And there are differences with age. So as the kids age, they develop more diverse urinary microbiome. And it seems to be that it's mainly driven by increase of diversity measures for the boys population. It also seems to be, regardless of the age, the diversity of the urinary microbiome in girls is higher, and it remains throughout the life, as we know from other compositional studies. We also can now see and resolve with the so-called long synthetic reads sequencing methods, such as loop genomics method. We can now define species with a better certainty, which occupy boys and girls' urinary microbiomes, and start seeing the differences not only between individuals, but also abundance of different species per different sex. So for example, presence of Prevotella, Shale, and Anarococcus species, which are more prevalent in female patients. But then we know that with age, the population is changing, and adult female microbiome is quite well characterized now, thanks to the work of several people, including several groups present now on the call. And we know that it's very widely diverse, which means that during, if you compare different individuals at any given time, we know that it's very diverse, but we can try to group all the patients based on what kind of Lactobacilli, which is one of the dominant genera occupying urinary tract of females present. And some studies identify several types of groups that dominated by L. crispatus, Gassera, Jensenii, Rhamnosus, Iners, and other Lactobacilli species. So I'm showing the snippet of one of the recent studies by Dr. Naz Siddiqui from Duke University Group, showing the differences between populations of postmenopausal women who are either healthy in in respect of UTIs, or they were diagnosed previously with recurrency of urinary tract infections. And in that study by Dr. Siddiqui and Dr. Carson, they showed that there is some association with the Lactobacilli species present in the cohorts, those with the recurrent UTI. In this particular figure, only the Lactobacilli representatives are highlighted by color and shown with a relative abundance. And the only thing which I want to show from this comparison is that there is high variability of species, and it seems to be that only few species of Lactobacilli are usually present in each particular sample, and they seem to be associated with other healthy or recurrent UTI cohorts. So, but there seems to be, based on this study and few other studies currently, that there seems to be association between urinary microbiome and predisposition to the recurrence of urinary tract infections. And therefore, we may consider that presence of particular microbes in urinary tract may somehow affect or predispose patients to developing uropathogens. So with this long introduction, I want to kind of introduce the main aspect of our study, which I'm going to present today, where the urinary microbiome can resist or interfere with development of urinary tract infections. We are going to focus on one particular repository, which we collected with collaboration with Dr. Siddiqui's lab. During the study data of which I just presented, we were lucky to have access to the isolated microbes from the urinary bladder, from all the 3 cohorts of patients of those postmenopausal women, from those who did not have no UTI diagnosis, those which were diagnosed with recurrency, and those which were also put on prophylactic antibiotics. From that collection of several tens of different species, we have a small but what we think representative collection of the bugs which are usually inhabit the urinary bladder of those women. And we have 27 different strains with 7 different species, some of which you may already recognize based on a description of the predominance of Lactobacilli in female urinary tract. And if you're not microbiologist, you probably heard about Lactobacilli from a completely different perspective. Definitely not talking about the urine of postmenopausal women. We usually consider Lactobacilli as a friend of humanity, or they're generally regarded as safe microbe. Those are good bugs which we use for centuries to food fermentation, to prevent food from storage, to create nice cheeses, yogurts, different fermented goods. And you're very well familiar with that, and many of you might even have taken probiotics, which we use in a majority from different genera from Lactobacilli, because they consider it to be good for gut health, lung health, vaginal health, and of course now you can purchase quite a few probiotics which are being marketed towards the urinary health or urogenital health. When a huge number of Lactobacilli species also used in the feeding the livestock to prevent infections of those instead of antibiotics, not associated with any particular brand presented here. Oh, I do apologize. Something happened with presentation. Please confirm that you can still see the slides.
Hey, Tanya, looks like we can see it.
Okay, good, because it just disappeared from my screen. I do apologize.
Oh, no.
So, microbiologically speaking, the consideration of Lactobacilli as good bugs is actually indicating that they are secreting a lot of noxious stimuli. So they're producing chemicals which are inhibitory for other microbes. They acidify the environment, so they lower the pH by releasing lactic acid and other organic acids, and therefore they inhibit growth of many microbes. They produce hydrogen peroxide, biosurfactant, different bacteriocins, and other toxins, which are either directly killing other microbes or diminishing their growth. Therefore, they're actually good competitors. And the presence of them in the field was previously considered to be explaining the protective effect in, let's say, vaginal space, where outgrowth of Lactobacilli thought to acidify the media and therefore basically expel anybody else, including different pathogens, therefore serving a protective effect. And from microbiology speaking, presence of Lactobacilli in urine tract, we can consider the question whether these good guys can resist the infection or presence of uropathogen. Official term would be colonization resistant. But for that, because these Lactobacilli are not isolated from yogurt, they're not used for millennia, we want to figure out what the genotype and phenotype of those bacteria, whether they can interfere with urinary pathogen, and that would be the main focus of today's presentation, and whether they can interact with other urinary commensal inhabitants, microbes. So one of the methods I'm going to be showing is taught usually in general micro classes, and that's how we test different chemicals affecting the growth of bacteria. For example, using inhibition assay, otherwise called well diffusion assay, and so forth. When we have growth of particular bacteria, we add different test solution, which can be antibiotic, which can be other bacteria, and then we observe whether bacteria around this disc or well are growing. And if they're not growing, we have an inhibition zone or clearance zone, and then we can discuss like why and then not growing there. So what happened there? In the context of trying to test the interactions between uropathogens and bacteria which are already present in our bladders, we can basically change this assay. And that's what we did initially. And we tried to load the lone or confluent growth of the uropathogen and then test how different lactobacilli culture and their derivatives are affecting growth of the pathogenic bacteria. And in reality, the plates look something like that. When we have control, we grow a non-pathogenic strain of E. coli, and then we're testing the bugs coming from the patient. So they clinical isolates from either healthy or diseased individuals, and then observing the clearance around those wells and quantifying it using imaging software. So, and then we can produce basically the data, which I'm going to be showing in the next few minutes. So first, we had initial isolates which we sequenced back in 2018. In 2019, we started testing them, obtaining the graphs like that. So if you take different strains coming from two different patients, 5006 and 5010, L-Gasseri and Delbruecker, we can then test them against pathogenic bacteria. No, not working with domesticated E. coli, but actually those used in models, and you may recognize some name of the UPEC strains such as SFT, UTI-89, Klebsiella pneumonia, and Fificalis being the major pathogen, and then test whether the urinary lactobacilli can inhibit growth of those using well. So what we observed in this initial set by testing two different strains of lactobacilli, that the majority of them inhibit all the three different pathogens which we tested, including not only gram-negative bacteria such as E. coli and Klebsiella, but also gram-positive such as Fificalis, which is much harder probably to kill. And we also noticed that there are differences in strengths of inhibition, and through the physiological development of a culture of the lactobacilli, the strengths of inhibition may change for some of the interactions significantly. And at that point, we were checking the patient's information about where the source came from this particular lactobacilli isolates. And we observed that L-Gasseri came from the healthy cohort, while L-Delbruecker, which is not a good inhibitor, as you may see from the right graph, came from the recurrent UTI cohort. And we, at that point, come up with very simple hypothesis, which I'm now thinking was quite naive, even though I still like it. We decided to test whether that can explain, or at least come closer to explaining, why recurrent UTI patients might be more predisposed if they have weaker inhibitor lactobacilli inhibiting their urinary tract. So we expanded our screen to all of the lactobacilli which we isolated from catheterized urine and which can grow consistently under the same conditions, so trying to basically have a very standardized test. And we tested the 19 different species of lactobacilli and some other urinary isotope lactic acid producing bacteria such as Strep anginosus. And here's the result testing this collection of lacta against the CFT73 UPEC strain, showing that majority of those different species and different strains of the same species which we had can inhibit E. coli to some extent, majority of them. But it's very variable, and sometimes even within the same species, we have different strengths of inhibition. We conducted the same screen against other uropathogens such as Klebsiella and Fificalis, and observed similar to that, majority of them would be inhibitory, but it's much harder to kill gram-positive uropathogen. So, but then coming back to the hypothesis, whether those isolates coming from the recurrent UTI patients can be actually bad inhibitors, we marked all the three cohorts, so no UTI, recurrent UTI, and those on prophylactic antibiotics, and we observed no significant correlation. So we were quite sad. We tried to quantify it different ways. So at that point, we only have a couple different strain representative from each species. So we lumped them all together and measured whether the lactobacilli coming from which cohort and plotted it against different pathogen. And you can see result here, there might be some variability. There's no significant change between cohorts, which make us think that our hypotheses are originally not correct, and it cannot be. UTI is a very complex process, as we all know, and it cannot be just reduced to the presence of what kind of lactobacilli present. In addition to that, we do need to expand our collection. We need to work, and not only with isolates which are coming from the urinary tract of postmenopausal women, which are clinically known to be more predisposed to recurrence of urinary tract infection. And therefore, we need to continue working to basically make a better conclusion in investigation whether lactobacilli can have a potential for the resistance towards UTI. We also work in trying to figure out what is the mechanism of the inhibitory action. So in this case, we have some snippet of the mechanistical study trying to figure out what noxious chemical, in this case, play a role in urinary lactobacilli isolate, showing that L-gasseri seem to be having a combination of different inhibitory mechanisms, which either relying on presence of lactic acid plus additional substances which are not proteinaceous based on the heating inhibition here. And L-dilbruca seems to be very narrowly inhibiting using the acidification of the media, in particular type of the culture, so during a certain physiological state of this particular bacterium. We also know that this inhibition is not specific to the surface, so it may work within the urine environment, or at least work in vitro in liquid media when we co-culture in different combination of the uropathogen and presence of lactobacilli. Based on the ratios, it may strongly, either completely or strongly inhibit growth for L-gasseri, which has a combinatory inhibition mechanism, and can only slightly decrease the proliferation of UPEC strain in the liquid if we have the combination of L-dilbruca at different concentrations. So we continue working on that, but the major summary from this part, and I'm sorry it took so long with interruptions, we summarize that majority of urinary lactobacilli from the humans, they behave similar to other lactobacilli isolated from different sources, because they inhibit many other bacteria, including uropathogens. And that inhibition is not dependent on whether the uropathogen is drug resistant or not, which is providing us with a hope that potentially that may be an intervention strategy towards drug resistant infection. And we see a lot of differential diversity between different strains and species. So it's not any lactobacilli which will be doing that, and therefore we need to explore further the genetic and phenotypic differences in different lactobacilli. So with that, coming kind of to the second short story I'm going to present today is that we're thinking that urinary microbiome is complex. We know that now from multiple studies, and that maybe these binary interactions just don't recapitulate all the complexity of this ecological niche. And of course, in this particular in vitro study, we completely ignore the effect of the host in other parts of the microbiomes and the complexity of development of this infection. But we, with the initial data that in vitro, the lactobacilli can resist the growth of the uropathogen, we are hopeful that we can try to work towards development of urinary tract specific and more designed probiotic, which would modulate the urinary microbiome or other microbiome specific way. So basically, let's try to get more information so we can have some informed development of the diagnostic and prophylactics and potentially treatment. There are books written about the topic, what creates a good probiotic, but unfortunately, it's still pretty much sparse science. Again, not association with any of the example, just to kind of, you probably all seen these bottles of probiotics which are on the market. But in general, the whole idea of probiotics comes the idea that it should inhibit or prevent growth of the pathogen. It should survive in particular niche, or at least colonize it temporarily. And it should be able to deliver to the niche because, for example, being in urological health niche, we want to make sure that we have some probiotics which can be given orally and probably not through the bladder installation, though it's still a possibility in some cases. Coming back to the data which I already showed, I want to draw more attention to the abundance of the specific lactobacilli species within the different patient environment. We only see that in each patient, we see up to few, let's say, or even sometimes just one lactobacilli present. And considering the idea that we're trying to modulate this microbiome and rely on a good inhibitor lactobacilli to prevent infection, we would like to know that whether a particular strain can survive within this niche and can be stable and successful. And from microbiological perspective, we know that lactobacilli are the best inhibitor from like bacteria because that's what they usually compete most strongly against. And the very brave master student in our group, Haley Hinze, conducted the initial interbacterial screen to test lacta versus lacta inhibition. So lactobacilli, as many of you know, are not the most happily growing in the lab bacteria. Some of them are more or less sensitive to the presence of oxygen and so forth. So Kaylee screened 6 different species, and 5 of these interactions are presented here. She found out that L. rhamnosus, one particular strain isolated from this patient, inhibit very well Ehrlich Brucker strains from another patient, and has minimal but measurable inhibition of other 2 species, such as Johnsonii animalis, but cannot significantly inhibit growth of the gasseri, which is abundant in some of women. We then took other L. rhamnosus strains isolated from other patients and tested them against the same set of other urinary commensal. And she observed that rhamnosus strains coming from other patients or coming from the other source of isolates, so coming from ATCC. And they actually able to well inhibited Ehrlich Brucker and animals. And then she also tested that this inhibition seems to be an unusual one to some extent because it's cell-dependent, or at least it has to be triggered by interaction of these 2 microbes, and it doesn't rely on just acidification of the media. And if she takes all secreted components, so just take cell-free filtrates of the culture of rhamnosus, it loses all inhibitory power, right? So there are different hypotheses we're going through how to identify this, but additionally, we wanted to confirm that it's not just one-off interaction. If we take Delbruca strains, which we have coming from different patients, they also all can be inhibited by this set of different rhamnosus strains, which makes us very hopeful that potentially we can identify this kind of interactions. And we continue to expand this to test different lactobacilli species and different strains, and we're looking for more. So if you have some nice lactobacilli collections, we would like to work with you further to establish broader screen, right? And basically find good competitors for growth of lactobacilli as well as growth of uropathogens, like a combination of 2 screens which are presented today. We're working to try to establish mechanism of inhibition, and we're assuming that for different species, we'll observe different mechanisms. And we're doing initial genetic analysis of the inhibition and, of course, of preference lactobacillus strain, coming from the idea that if you want to have a good urinary health probiotic, it would be nice to not constantly take probiotics, but colonize the bladder of a particular individual with a strain which can then resist development of UTI. To summarize this small screen part, studying interaction of what's present already in the bladder without presence of any uropathogen, we can conclude that urinary lactobacilli specifically interacts among themselves, in this case, species specifically, and these interactions are potentially very important for development of probiotics and treatments towards the urinary tract infection. To summarize all of this story, and I again apologize for interruptions, we see that urinary tract is home for complex microbiome. It develops throughout the lifetime of an individual, and it has potential to interfere with development of infection, which we can study further to develop interventions strategies. And so we're hoping that further characterizing urinary commensals, in particular lactobacilli, we will be able to work towards developing new antibiotic-resistant bacterial infection interventions. So basically hoping to develop new strategy to diagnose predisposition to recurrence of urinary tract infection, to figure out the strategy to produce probiotic, which can be either prophylactic or even curative towards the UTI. So with that, I would like to thank many people who contributed to development of this project, including our students, of course, our collaborators, some of them present on the call. Thank you for support. And of course, big shout out to our funding sources, including CAIRIBU grants, and of course, to all the supportive community who make this research possible. Thank you. And I will take any questions if you have some.